Hello everyone, welcome to Biotech Notebook. In this video, we are going to see the overall view about fermenter. So this video covers what is fermenter, parts of fermenter and its function, then the important parameters which you have to monitor and control during your fermentation process. So fermenter is a closed vessel which provides a controlled environment for the growth of microorganism or animal cells to obtain your desired product. Okay. So this is your fermenter, which is a closed vessel which provides a controlled environment for the growth of the microorganism for producing a desired product. You have to design your fermenter mainly based upon the type of process and the fermentation product which you are going to produce inside the fermenter. Let's see the size of the fermenter. Okay, so in fermenter you have two types in size wise, that is laboratory scale fermenters and industrial scale fermenters. Okay, laboratory scale fermenters are in small size, that is 1 to 20 liter. Then industrial scale fermenters are in large size, they are 1 lakh to 5 lakh liter or even more big size of fermenters are there. Then one more point you have to note here is generally 20 to 25 percentage. Okay, so 20 to 25 percentage of fermenter volume is left unfilled with medium as head space. Okay, so this head space is for splashing, foaming and for aeration. So only 75 to 80 percentage, okay, of fermenter volume is filled up with liquid. So if you consider this is your total volume of your fermenter, okay, in which you will fill up with only 75 to 80 percentage. Okay, 75 to 80 percentage of volume is filled up with your liquid. Okay, so liquid included your nutrients, that is medium, then your inoculum. If foaming will occur, you will add anti-foaming agent during your process, then you may add acid or a base to control the pH. So everything will include in this 75 to 80 percentage. Okay, remaining 20 to 25 percentage is known as headspace volume. Okay, so this is your headspace volume. This headspace volume is for splashing, foaming and aeration. So this is your working volume and this is your headspace volume. Next one is construction material. In fermenter with strict aseptic requirements, it is important to select materials for the body of the fermenter. So you have to select the body of the material, that the material should withstand repeated steam sterilization cycle. Okay, so whenever you are starting fermenter, you have to sterilize the fermenter to kill or destroy the microorganism present inside your fermenter. So your fermenter should withstand repeated steam sterilization cycle. For that you have to select the good construction material for the body of your fermenter. Okay, small scale fermenter you will use glass or stainless steel. Okay, so in which glass is useful because it gives a smooth surface and it is non-toxic, then corrosion proof, then it is very easy to examine the interior of the vessel. Okay, so you can easily view the what is going inside the fermenter if you use glass container for your fermenter. Then you have two basic types in your glass fermenter. One is a glass with round or flat bottom and top flanged carrying plate. Then second one is a glass cylinder with the stainless steel top and bottom plates. Okay, here your top and bottom only made up of stainless steel and your body is made up of glass. This model you can sterilize by autoclaving method. Autoclave is a steam sterilizer, autoclave is a machine that uses steam under pressure to kill harmful bacteria, virus, fungi and spores on items that are placed inside the pressure vessel, that is your autoclave. So you have to keep object or your vessel inside the autoclave machine for sterilization process. And in second model method you can sterilize in situ, but 30 centimeter diameter is the upper size limit to withstand the working pressure. Okay, then when you consider the cost of the fermenter, the second type, that is vessel with the two stainless steel plates, cost approximately 50% more than that of with a just one plate. In large scale fermenter, all fermenters are sterilized in situ. That is, you will sterilize your fermenter without taking it apart. That is, you will sterilize in the same place. Okay, so any materials used will have to be assessed on their ability to withstand pressure sterilization. Okay, so this material should withstand pressure sterilization, then corrosion on their potential toxicity, then the cost should be minimum. Large scale fermenters are made by stainless steel or at least have a stainless steel cladding to limit corrosion. Okay, next, let's see parts of fermenter and its function. Okay, so first the main important structural component of fermenter is agitation and aeration system. Okay, so this aeration and agitation system consists of the agitator, that is impeller, then stirrer glands and bearings, baffles, then aeration system sparger. Okay, so agitator also known as impeller, then aeration system is known as sparger. This is your agitator, so it has motor on head of it, then it has a shaft, then this is stirrer shaft seal, and this is your impeller. So impeller may be single bladed, or in some fermenters it may be multi bladed. Okay, so depending upon your process you can select single bladed agitator or you can select multi bladed impellers. So this is your agitator setup. So mainly this agitator is used to mix your bulk fluid inside your fermenter. Okay, so bulk fluid and gas phase mixing, then to disperse the air, then oxygen transfer, heat transfer, suspension of solid particles present in your fermenter, then to maintain a uniform environment throughout the vessel content. Okay, then it also used to break the air bubbles produced during your process in the liquid medium. The next one is you have different types of agitators, so which are known as disc turbines, vanned discs, open turbines of variable pitch, and propellers. The next one is stirrer glands and bearings. So this is used to seal the openings of a bioreactor. Okay, this is used to seal the openings of a bioreactor. So this is used to restrict the entry of air from the outside. Okay, so tight sealing restrict the entry of air from the outside. Okay, the stirrer shaft, it may enter the vessel from the top side or bottom of the vessel. Okay, top entry is most commonly used. Then bottom entry may be advantage if more space is needed on the top plate for entry ports. Okay, suppose if you need more space for entry ports, like ports for adding nutrients and adding antifoam agents, then for acid or alkali for pH control. And there are three types of stirrer glands and bearings. The one is stuffing box and mechanical seal and magnetic drives. And the next one is baffles. 4 baffles are incorporated into agitated vessels of all sizes. Okay, these baffles are mainly used to prevent a vortex and to improve aeration efficiency. Okay, so these baffles are nothing but it is a metal stripes roughly 1/10 of the vessel diameter. So this is your baffles. So the size should be 1/10 of the diameter of your fermenter. Okay, these baffles are attached radially to the wall. There should be a gap between baffles and the fermenter vessel wall so that there was a scoring action around and behind the baffles, because which minimizes microbial growth on the baffles and the fermentation walls. Okay, then if you want extra cooling coils, you may be attached to the baffles to improve the cooling capacity of the fermenter, because you have to maintain the temperature of the process. Okay. The next one is aeration system, that is sparger. Okay, a sparger is a device for introducing air into the liquid in a fermenter. So this is your air sparger, which is used to introduce air into the liquid in a fermenter. Then there are 3 types of sparger. One is porous sparger, then orifice sparger, and nozzle sparger. Next one is temperature control. When you design a fermenter, it is important to control the temperature your process. You have to maintain the optimum temperature for getting your desired product. During the fermentation process, heat can be produced mainly in 2 ways. It may be due to microbial activity or your biochemical reaction. Then the heat may be produced due to your mechanical agitation. If the heat generated by these 2 processes is not ideal for your particular manufacturing process, then maybe heat to be added or removed from the system. It's depending upon your fermentation process. Okay. In small scale fermenter, little heat is normally generated. So you have to give extra heat if the process needs heat. Then This extra heat has to be provided by placing the fermenter in a thermostatically controlled bath or by the use of internal heating coils, okay, or a heating jackets through which water is circulated, or you can use silicone heating jacket. So these all are used to heat your fermenter for obtaining a desired temperature of your fermentation process. Then in pilot scale fermenter, it is not possible to use silicone jackets due to the larger size of your fermenter. Okay, so in large scale fermenter, you may have internal heating coil which is used to provide extra heat, and cold water circulation helps to remove excess heat. Okay, if you want to heat your system, you can circulate hot water, or if you want to minimize the temperature of your process, then you can circulate cold water. Okay, so this internal heating coil is used for both heating or cooling the system. And temperature probe is used to measure and monitor the temperature changes in the medium or your culture broth during the fermentation process. Okay, so you can maintain your temperature of your fermentation process by using this temperature probe. So next one is foam control. So this foaming is occurred due to some component present in your medium or some factors produced by a microorganism during your fermentation process. Okay, so it is very important to control the foaming, or otherwise it will lead to some danger. That is, it may affect some physical and biological factors of your process. So the physical factors include reduction in the working volume of fermenter, changes the bubble size, then it lowers heat and mass transfer rate, then there will be invalid process data due to deposition of this foaming on sensor devices. So interference at sensing electrodes. So it leads to incorrect monitoring and control of your process. So it is very important to minimize foaming. When foaming becomes excessive, then there is a danger that your air filter becomes wet, resulting in contamination. So to prevent this foaming, we can use anti-foaming agents, or you can go with mechanical foam breakers. Foaming agents or cottonseed oil, linseed oil, soybean oil, olive oil, castor oil, sunflower oil, rapeseed oil, and cod liver oil. Okay, then silicones and sulfonates are also used as anti-foaming agents. Okay, so there will be a foaming probe to monitor the foaming, excessive foaming inside the fermenter. So if there will be excessive foaming, means how to add anti-foaming agents inside the reactor. So this is your anti-foam port. Okay, if your foaming sensor will detect that there will be excessive foaming, then you can add your anti-foaming agents to this port, or otherwise you can use some mechanical foam breaker. It's based upon the process you are working with. Then, like anti-foam adding port, there are some other ports which are used to add nutrients inside the fermenter. Then there will be a feed port for addition of acid or alkali to control the pH of the medium. Okay, so you have to add acid to neutralize the basic environment and maintain the required pH. Okay, then alkali is used to neutralize the acidic environment and maintains the required pH. So pH probe is used to evaluate and monitor the change in the pH of the medium. So this is your acid or base port, and this is your aseptic inoculation pipe, and separate port for adding nutrients, adding medium to the fermenter. In small scale fermenters, your feed ports are made up of silicone tubes. Then before starting the process, you should sterilize all your pipelines and fermenter vessels. So these silicone tubes are autoclaved separately, and your nutrients are pumped into a silicone tube by peristaltic pump after aseptic condition. Okay. The next one is sampling port. The sampling port is mainly for monitoring and analysis of the reactor broth condition. So this is your sampling point. In order to control and optimize your bioreactor function, so you have to frequent sampling of small volume of culture is required. Okay, so to optimize the function, you have to frequently take up the small volume of culture, and you have to evaluate different parameters. So later in this video, we will see what are all the parameters you have to monitor and control during the fermentation process. Then the sampling process should be designed in such a way that non-sterility should be provided before and after the sampling. So next let's see about valves and steam traps. So these valves are used to control the flow of gases and liquids in a number of ways. Okay, so there are different types of valves, or gate valves, globe valves, piston valves, needle valves, plug valves, ball valves, then butterfly valves, pinch valves, diaphragm valves, check valves, pressure control valves, safety valves. Okay, depending upon the type of fermenter and type of fermentation process, you can select any one of these valves. Next one is steam traps. These in all steam lines, it is essential to remove any steam condensate which accumulates in the piping to ensure optimum process condition. This may be achieved by incorporating steam traps. So steam traps are used to remove your steam condensate which accumulates in the piping. Okay, so these steam traps will collect and remove automatically any condensate at appropriate points in steam lines. Okay, so steam traps are used to collect and remove your steam condensate formed in your steam lines. And next one is the various parameters you have to monitor and control during your fermentation process. So the physical parameters are temperature, pressure, reactor weight, liquid level, foam level, gas flow rate, medium flow rate, culture viscosity, gas holdup, and power consumption. Then your chemical parameters are pH, dissolved oxygen tension, dissolved oxygen concentration, redox potential, then exit gas composition, conductivity, broth composition. Then biological parameters are biomass concentration, enzyme concentration, biomass composition, then viability of the cells, then morphology of the cells. Then in the next video we will see how to measure these parameters and why we have to monitor and control these parameters during your fermentation process. Thank you for watching my videos. If you like my videos, please like, share and subscribe. Thank you.